Okay, I'd like to welcome you to this afternoon's discussion about the genetics of bitter taste perception. And one of the people who's going to be introducing this topic to you is Dr. Michael Campbell, who is a postdoctoral research fellow in my lab at University of Pennsylvania. Michael actually started out with an undergraduate degree in anthropology, I believe, and then did a PhD at Columbia University, also in anthropology, but he also did research in a lab because he knew he was interested in getting that sort of experience. And then after he finished his PhD at Columbia, he wanted to get additional experience in doing human genetics research and learning about population genetics analyses, and so he came to my lab at Penn. And while he was at Penn, he was actually the one leading the study of bitter taste perception. Michael?
Hello, everyone. Taste perception starts on the tongue and soft palate, where specific chemicals in foods and drinks interact with taste receptors found on the taste receptor cell. Now, taste receptor cells are grouped together to form taste buds across the surface of the tongue. And each cell within the taste bud has the ability of responding to one of the 5 elemental tastes, namely sweet, sour, salty, bitter, and umami. Now, here's an enlarged picture of the taste bud on the left-hand side of the slide. And on the right, we have an example of a bitter tasting chemical interacting with the bitter taste receptor, which is on the surface of the taste receptor cell. And the taste receptor cell forms a synapse with a nerve that sends electrical signals to the brain where bitter taste is perceived. And now for some audience participation. I believe that you've all received an envelope. If you'll open that envelope and taste what's inside, we can determine your level of sensitivity to the bitter compound PTC. Okay, so go ahead. I love these expressions. That's the strongest one that I saw. No, that one. Yeah. It's very strong. Yeah. Oh, man. It's not that bad. It's PTC, phenylthiocarbamide. Really? You don't taste it? Okay. All right. By show of hands, does anyone taste this chemical? Who can taste it? Okay. Yeah, okay. Who does not taste it? Yeah, okay.
I would estimate that roughly 75% of you were able to test. It was almost like classic, like 25% couldn't and 75% could. And so one thing to think about, remember there are 2 common variants, right? And think about the different combinations you can have. So you could have 2 of the, taster and be heterozygote or not. And one thing we should mention is that the taster haplotype is dominant. Yeah. So think about that and think about why you would get a 75 to 25 ratio.
Okay. The ability to detect PTC bitterness is often cited as a classic example of a highly variable trait in humans. Individuals who find PTC bitter are classified as tasters, and those who find PTC tasteless are considered non-tasters. Now, the ability to taste PTC is also correlated with the ability to taste naturally bitter substances in food. PTC has been used in taste perception studies to understand the link between genetics and taste perception. Now, dietary sources of bitter substances include vegetables such as broccoli, bok choy, cauliflower, cabbage, watercress, etc. And these foods contain bitter-tasting phytochemicals, as well as antioxidants, fiber, vitamins, and minerals. Although bitter substances are present in beneficial foods, many toxins in plants are also bitter. So it's been hypothesized that bitter taste perception likely evolved in humans to prevent the consumption of potentially toxic chemicals and may represent a defense mechanism in our species. Now, the bitter taste receptor gene TAS2R38 on chromosome 7 was discovered in 2003. And this gene accounts for a large proportion of the phenotypic variants in PTC perception. This gene consists of two untranslated regions depicted here in blue and a 1002 base pair coding region depicted here in red, which encodes for a 333 amino acid receptor protein product. 3 SNPs within the coding region of the class 2R38 gene have been found to explain up to 85% of the phenotypic variance in PTC sensitivity. Now, these polymorphisms can cause amino acid substitutions in the 333 amino acid receptor protein. For example, variants at nucleotide position 145 encodes for either a proline or an alanine, and those are 2 amino acids. Variants at nucleotide position 785 encode for either a alanine or valine, and variants at nucleotide position 886 encode for either a valine or isoleucine. Now, the different combinations of these amino acids in the receptor protein can give rise to different amino acid haplotypes. For example, when proline, alanine, and valine occur together in the same receptor protein, this particular combination is known as a PAV haplotype. In our recent analysis of the TAS2R38 gene in Sarah's lab, our sample consisted of 611 individuals from 57 African populations, and these populations practice diverse modes of subsistence such as hunting and gathering, agriculture and pastoralism, and they speak languages belonging to the Nilo-Saharan, Niger-Kordofanian, Afroasiatic, and Khoisan linguistic families. As well, we included 132 non-African individuals from Europe, the Americas, Middle East, South Asia, and East Asia for comparative analysis. Now, non-Africans, as you see on the right, have 2 common haplotypes, namely PAV and AVI, which constitute about 99% of the sample chromosomes. Also, among non-Africans, we found a low-frequency AAI haplotype. In contrast, among Africans, we found that PAV, AVI, and AAI occur at high frequency, and as well, a low-frequency AAV haplotype was observed among Africans. Bitter taste phenotypes were also measured in 463 individuals from our 57 populations. A PTC test was administered to these individuals using serial dilutions of PTC in vials labeled from 13 to 1, with vial 13 containing the most dilute concentration of PTC and vial 1 containing the most concentrated solution. Now, individuals would taste successive concentrations of these dilutions, beginning with the most dilute, so they would begin with vial number 13, and they would continue to taste until they were able to detect the bitterness of PTC at a particular vial number, which would then become their score. And in this photo, we see 2 members of my lab, Alessia and Jabril, administering the PTC test among the Yaku people of Kenya. Here again, we can see my colleague, Alessia, administering the test to the Amolo people of Kenya. And this test is very time-consuming and it's laborious as well. And you can see the large number of bottles that are required to complete this test. In this genotype-phenotype plot, the y-axis is PTC response, which is basically the PTC score that was tested in the field. And genotypes are listed at the bottom of each column of bubbles. So you can see them along the bottom. And also, the size of the bubbles is proportional to the number of individuals with a given PTC score. Our results indicated that PAV individuals, and those are individuals at the extreme left of this plot, have a higher sensitivity to PTC compared to AVI individuals at the extreme right of this plot. Interestingly, we also found that there were a number of genotypes here that are associated with intermediate bitter taste response, and this is something that we really do not see outside of Africa. Now, some time ago, many of you participated in our own genotype-phenotype analysis. Specifically, 59 of you submitted saliva samples, and from the saliva, we extracted DNA, we amplified the DNA using PCR, And then we proceeded to analyze the DNA further using SNP genotyping technology to determine the specific alleles present at the 3 positions that I mentioned earlier that are associated with PTC sensitivity. And these are the results, or an example of the results. When looking at this information, it's important to remember that we each have 2 copies of genes in our genomes, one from our mother and one from our father. So at nucleotide position 145, individual 1, for example, has a CC. 1 C came from the father and 1 C came from the mother. This maternal-paternal combination is referred to as a genotype. And from the genotype data, we were able to determine the amino acid haplotypes for each individual. On the left-hand side, we have haplotype frequency, and we can see that PAV and AVI haplotypes occur at high frequency among the 59 individuals that we genotyped. We also observed 2 low-frequency haplotypes, namely AAI and AAV. If you look at the right, you will see the genotype frequencies, and we found that PAV individuals, PAV-AVI individuals, and AVI-AVI individuals also occur at high frequency within this sample. Now, the 59 genotyped individuals also performed a PTC test using taste strips like the ones that we used in our demonstration. An individual response was recorded as no taste, weak taste, or strong taste. The y-axis is PTC score. Genotypes are listed at the bottom of each column of bubbles, and the size of the bubbles, again, is proportional to the number of individuals with a given PTC score. And we can see here again that PAV individuals have a higher sensitivity to PTC compared to the AVI individuals, which is what we would expect to see. Here we have a family. We have a pedigree. Here, circles represent females, squares represent males. Across the top, the 2 colored circle and square represent parents, and on the bottom, from left to right, you have the son, the daughter, and the nephew. And it's interesting to see that if we look at the daughter, she's the green circle at the bottom, she received an AAV haplotype from her mother and a PAV haplotype from her father. And again, with the son at the extreme left at the bottom, he received a PAV from his father and a PAV from his mother. So this slide just basically demonstrates the inheritance of bitter taste haplotypes. Interestingly, we also found that in terms of the phenotypes, they're indicated here by weak and strong. The son, who has the PAV genotype, has a weak response to PTC. As well, the PAV-AAV mother has a weak response to PTC. Typically, we would expect to see a strong response just given their haplotypes. And basically, this is an example of how there is not necessarily 100% correlation between genotypes and phenotypes. There are other factors, environmental and genetic, that can also influence your ability to detect PTC. So what conclusions can we make from our data? Well, the widespread distribution and high frequency of these common haplotypes across diverse populations suggests that these variants are likely very old, and they probably predate the origin of modern humans. As well, we see occurrence of the AVI haplotype at high frequency across diverse populations as well, and this suggests that this particular haplotype is likely functionally important, although that function has yet to be determined. So these patterns, together with other patterns that I haven't mentioned here today, are consistent with a scenario of natural selection maintaining high levels of variation at this locus in modern human populations. And with that, I will stop here and answer any questions if you have them. Yeah.
I was wondering if the same groups pertain to the different types of taste. So are there maybe certain people like non-tasters and tasters for the sweet category or the sour category?
Yeah, I mean, in the literature, they suggest that if you have a greater sensitivity for detecting bitter, you have a higher sensitivity for detecting sweet. I mean, there's this correlation between the two. So
But I believe there's different receptors, right?
they're totally different
For
receptors.
sweet,
So
and there is
yeah,
variability.
right.
Absolutely.
But there's a correlation, but their underlying genetics is different.
Can taste actually connect with smell?
Yeah. Yeah, they're connected together. So part of the tasting process does incorporate olfaction as well.
And there are receptors, there are olfactory receptors, and they get triggered off as well. And there are different pathways in the brain, but there is supposed to be some connection between them.
I was just wondering, there's this like magical fruit, right, that blocks the bitter taste that makes it sweet. So if only certain people can detect this PTC type thing, is it also true that only certain people that magical fruit would work, or does it work for everyone?
Whatever the chemicals are, that is really weird. You're talking about this magical fruit. I read about it in the New York Times, and they're having like these magical fruit parties. Sounds like something really illicit. But in fact, all it does is, I guess you eat it, and then stuff that was sour suddenly seems
It's
sweet,
really sweet.
and things like that.
Yeah.
I have no idea what the chemical basis of that is. So again, there's probably, remember that this gene and this interaction, this is just one very specific example, but there are many different types of receptors.
Yeah.
These receptors exist in gene families. So there are clusters of these genes, and how many are there altogether? There's like
25 functional.
25 of the bitter taste
functional
receptors. And then you could imagine having different combinations of them could play a role in how you taste things.
Gene interaction. It potentially could be a very complex system.
And there are other things besides PTC that might play a role for this particular gene. And in fact, some people have hypothesized, they've said, well, why is there such a high frequency of the non-taster? Like why, what's the reason? We actually don't know, but some people hypothesize that maybe that version of the protein plays a role in detecting another substance. And we just haven't found it yet.
Right.
We just don't know what it is.
Yeah, the literature has been suggestive in terms of what that substance is, but there hasn't been any clear evidence. Okay, the girl at the back,
I was wondering when you said that umami was one of the 5 tastes, but originally everyone like years ago said there was only 4, so wasn't umami just recently discovered, right?
Yeah, umami is that savory taste in meat.
Or do you think there's like any more of them that we might discover?
That's an interesting question. I mean, potentially, why not?
My understanding is I'd be surprised, but you never know. It's possible that they'll find another, other types of perceptions. Yeah. But as far as I know, those 5 are the main ones.
I've heard different comments about whether or not where on the tongue something goes affects if you can taste it. How did you deal with that? Because is it true, first of all? No, it doesn't affect it. So it wouldn't have an effect on whether someone would say strong or weak sense of bitterness.
And also the way we do the tests in the field, they're kind of taking the whole substance and swirling it around. So that also makes a difference.
What's the typical number for like the sampling amount? Does it vary?
When we were in Africa, well, samples. Well, she's got thousands.
We tend to overkill a little bit. We do a lot of sampling, and it depends on the questions. So your sample size when you're doing an experiment, for example, it's really going to depend on the questions. And for human population genetic studies, typically people say they want a minimum of 25 to 30. But if you're trying to link genotype and phenotype, the larger the sample, the better. And ours was unusual because we looked at so many remote populations and huge sample sizes. What was it? In several hundred, right?
Yeah, 611.
And we had phenotype information, and then Michael collected the genetic information as well. And so you get more statistical power the larger your sample sizes are.
Yeah. And there were other things that we found when we were doing our analyses, things I didn't mention. We found rare variants that we never would have found if our sample size was smaller than it is. So, I mean, sample size, the larger the better. It's always good.
I was wondering if you noticed more like people had PTC testing in areas where there were poisonous plants.
You go ahead. You did the testing, not me.
So I think it's a really good question because it raises this question of what's the role of this? And so it actually had been hypothesized that the reason that we have this ability to taste bitter substance is because of toxins, poisons in plants. And so the idea is, well, you better know, which ones are poisonous, and if it tastes really bad, you're not going to eat that one. And so we purposefully, what's the example again in West Africa with the substance that Fatima Jackson studied?
Oh, cassava, the cyanide in cassava plant?
Right, and isn't that bitter, causes a bitter taste as well?
Yeah.
I mean, there are
absolutely.
correlations, different substances that people eat in different regions that can be correlated with diet. But in fact, that was the motivating force for us to do the study. And when we started out, we had a hypothesis. And the hypothesis was that if we look at all these different diverse African populations, and they have really different diets, and some of them are hunter-gatherers, and some are pastoralists, and some are agriculturalists, we expected we're going to see lots of variation. And we thought that it's because they would have different toxins or, in the plants that they're eating. And we found completely the opposite. Yeah. We actually found that they were remarkably similar. So although there's more diversity generally in Africa amongst all these different populations, and we're talking pygmy hunter-gatherers in the rainforests in Central Africa and East African pastoralists and many different groups showed a remarkably similar pattern.
Yeah.
So there goes that hypothesis. Yeah. Okay, so what's going on here then? Why are they showing such a similar pattern? So the other possibility is that the different variants are being maintained at similar frequencies in different populations for some other reason. Mm-hmm. And we don't know exactly what it is, but when you have this maintenance of different variants in populations, that could be due to something called balancing selection. So I gave an example in my lecture where I talked about the sickle cell trait, sickle cell anemia. And that tends to be common because, as I said, people who have one copy of the sickle allele and one copy of the wild type have protection against malaria, so it rises to high frequency in West Africa. Okay, and the maintenance of the different variants is called balancing selection. So the idea is that there could be a selective force out there that is maintaining these variants of that gene, TAS2R38, at high frequency in every global population. And we have no idea what it is. So Michael, do you want to talk about some of the possibilities of what role?
Well, I mean, recently it was found that on the lung you have the expression of these bitter taste receptors. And also the bitter taste receptors are expressed in the gut. So it's been hypothesized that they have these other roles that are not necessarily related to diet, but potentially they're related to human health. The reason, I hypothesize that these haplotypes occur at high frequency across these very diverse populations because they're very important at a very fundamental level. They're very important to human survival. So I'm thinking at the level of health, something really, really basic.
In fact, tell them how old. You inferred the age of these different variants.
Yeah, so you learned about coalescence in Sarah's lecture. And so basically we took that method, the coalescent method, and we looked at what point do all of these variants coalesce to a common ancestor? And we found that they coalesce at a time that's approximately 2 million years ago, which is about the time of the genus Homo. So we suspect, of course, we know that this gene is very old, but the variants that have emerged from this gene probably emerged they started to emerge after the origin of the genus Homo. And so you see all of this diversification
and
at that point.
before the origin of modern humans.
And before modern humans.
What was the finding in Neanderthals? Neanderthals had those,
and then
right?
Neanderthals also had one of the variants at nucleotide position 145. So they found a P and an A, basically. So they're hypothesizing or concluding that, yes, Neanderthals could taste, and they also had non-tasting ability as well. So that suggests that most likely that this variant is in the ancestor of humans, modern humans, and Neanderthals. So it's very, very old.
Now, there's one interesting thing that when we were in Africa and we were doing those tests, we ran across some people in Kenya. There were a handful of people, not a huge number, and they were what we call bitter blind. So we gave actually a series of different chemicals that are usually perceived as bitter. I think we gave 5 different chemicals, and each person would do that series of dilutions. And these people, you would get more concentrated, more concentrated, more concentrated, never tasted bitter for any substance. And they are what we call bitter blind. It's extremely rare. It has been reported. And there has to be, it can't be any particular gene that's defective. It has to be a more fundamental processing thing going on. And they would also say, oh, for example, I knew, there was something weird going on because when I go to get medical treatment, like for malaria, anti-malarial medication actually often has quinine, and that's another substance that some people taste as bitter and others don't. And he said, well, I never had any problem. I go to the hospital, I get treatments and medicine. I had no problem. My family couldn't stand it. And so we're actually interested in following up on those individuals and their families and maybe looking at pedigrees and see if we can identify the genetic variants that play a role in that trait.
Yeah. Now, the interesting thing with these receptors is that they also interact with a G protein on the inside of the cell. So the receptors on the outside, it couples with the G protein. And one potential explanation is that there's a defective G protein and you're not getting that signaling. So you can hit them with bitter substances and they're just not detecting them, even if they do have a receptor that's associated or that would normally allow them to detect bitter substances. So there's something else going on.
Like the downstream processing.
Yeah, like that signaling is just not going on.
Right.
I'd just like to inquire about human evolution. This may be an ideal, but is there a way that you can accurately calculate a rate of evolution, or does it merely, from a genetics point of view that is, or does it merely hold true to the nature and evolving out of necessity rather than having a direct correlation with natural evolution in a constant rate?
You're talking a bit about this molecular clock idea, like is there's constant ticking, and the answer is yes and no. People have shown that for mitochondrial DNA, for example, it's an approximation, it's not perfect. There are many factors that influence that clock, and keep in mind that the way we set and determine that clock is we look at modern day variation. What they will typically do is they'll compare the variation in modern populations to chimps, and then they'll say, or chimpanzees will say, okay, we diverged 6 million years ago, so what's the rate of mutation? But lots of things can affect that, like natural selection.
Yeah.
So that will totally mess up your clock. So if there's selection acting, that clock might tick really fast or it might go really slow. And so it can actually be kind of complicated, but it's not always a perfect clock. We do our best to approximate that.
I wanted to know if you guys have tried the PTC experiment with other animals besides us humans.
Well, it's been done with chimpanzees. And it was found that they too have a taster phenotype and a non-taster phenotype. Whether intermediates, that wasn't discussed in the paper. But yeah, apparently the taster phenotype is ancestral. So, I mean, that's basically, chimps aren't tasters. Our ancestral trait is tasting. But then for some reason, non-tasting evolved in chimps and non-tasting evolved in humans independently. So the variant that's associated with non-tasting in chimps is not the same variant that we see in humans that's associated with non-tasting. They're completely different, but still the trait evolved twice. So
Can they taste PTC?
yes.
So how do you know they can?
Oh, that's a great,
Yeah.
I love this experiment. Do you want to tell them? Or?
No, you go ahead and tell them about the chimp experiment.
How do you test a chimp to find out if they can taste PTC? You take orange is what I thought they used actually.
Oranges or apples or something.
Right, they took
Some
a
kind of
fruit that they really like to eat and put PTC on these. And basically the chimps would go, ugh, yuck. They could taste it. You would know, apparently you knew immediately if that chimp could taste it or not.
Yeah.
And that's how they did it.
Yeah.
It was a really, really neat study actually.
Yeah. And there was a correlation between what they found in terms of the phenotype and the genotype. So, yeah.
Yeah.
Upon finding some sort of results that do show a connection between the genotype and the phenotype, what sort of conclusions can you make in the human's evolutionary pattern off of that?
Well, there are two things. Balancing selection is one of the conclusions that we made looking at the patterns of genetic variation at this gene.
And phenotype.
And also with the phenotype data, we also found these novel variants in Africa that have never been found before outside of Africa, and they modify the ability to taste PTC. And so this is an example of having rare variants that can actually modify your ability to detect PTC.
And if we didn't
bitterness.
have that phenotype, we wouldn't have been able to link these particular amino acid changes.
Right, exactly.
But another more general, we could relate this sort of to lactase actually. because it was very important to be able to link the genotype to the phenotype. In the case of lactase, to know something about what the selective force is and what selection is acting upon. It's really, some people consider it ideal. I talked about looking for signatures of selection in the genome, and I gave the example of lactase. And a lot of us are interested in looking at genome-wide. We'll genotype a bunch of markers and sort of look for those big regions where everybody's identical. And that might be a signature, okay, there's something important here. There's a functional variant somewhere in here. And it's actually been hypothesized that common diseases, diabetes and heart disease, for example, might be common in modern populations because they were selectively advantageous in the past. And so the idea being that we were all hunters and gatherers to about 10,000 years ago. So maybe having a variant that really quickly, was good for fat storage at the time, but in today's environment, it's no longer a good thing. And so the idea is if you could find those genes, use these signatures of selection to find those variants, it might tell you something about finding genetic variants that play a role in disease susceptibility. But let's say you did that. You search the whole genome, and I find some region, and it shows a signature. The key thing is for me to link that with a phenotype. Drawing those two together to show what is the function of that genetic variant. That's going to be the key, actually, and that's really, really hard to do. Yeah. It's a huge challenge that we all face, actually.
Yeah, and it's very critical for lactase persistence. With our stuff, we found that there wasn't very much variation in terms of sensitivity, whether you were a hunter-gatherer, a pastoralist, or among these different groups that practice diverse modes of subsistence. So we were able to sort of rule out, we still don't even know what is driving the high levels of variation at this particular locus. And so at least now we can rule out that, it's not diet, it's something else.
I was just wondering what some environmental factors could be for whether or not you can taste or not taste, because some of the genotypes were the same but different phenotypes.
Yeah. So you're getting this disconnect between genotype and phenotype. There are people that we have in our lab who go out into the field and who do these tests, and they feel that things like smoking can influence your ability to detect bitter substances. Also, there's this chemical that people in Ethiopia like to chew.
Okay.
Yeah, and it's been suggested that perhaps that can, in the short term, alter your ability to detect bitter substances. There are genetic factors as well that can influence your ability not to taste PTC or bitter substances, even if you have the underlying genotype that would enable you, or should enable you to detect these substances.
And just following up on that, that's because this is not entirely a Mendelian trait. And by a Mendelian trait, I mean a trait that is determined by a single gene at a single location. So that TAS2R38 gene, it accounts for most of the phenotypic variants, your ability to taste that substance, but not 100%.
Right.
So there's other genes out there. Yeah. And I think it's like 80% it accounts for?
Yeah,
something
85%,
like that?
so you're missing 15. So
Right.
there's...
So there are other genes, and they haven't found them yet.
Yeah.
They're still looking for those.
We'll find them.
I was just wondering if you found any specific examples, like of other genes that influence the PTC, like.
PTC specifically. No, not PTC specifically. We have not analyzed other genes that are related to PTC other than TAS2R38. But there are other bitter taste phenotypes that we have been looking at, like salicin, for example. And that's also correlated with a gene called TAS2R16. And so that's just another example of phenotype-genotype correlation as well.
So yeah, and these bitter taste receptors, they actually exist as families of genes.
Yeah.
They're like gene clusters. And they tend to be located in the same region of the genome. And you'll see a whole bunch of genes together, I think, maybe about 20 per cluster.
25. Yeah, okay.
And sort of end to end. And so that can account for a lot of the diversity in our ability to taste different substances. And so here we're just focusing on one very specific example, but there's potentially a lot of other substances and other genes that could be involved in perception.
Yeah, it's quite complex. You can have gene-gene interaction. You could have variants within genes that can affect your sensitivity. It can be potentially very complex.
Is
the reason why you chose the bitter taste because it's the only taste that can be found on the most of the phenotypic ratio can be found on one gene?
Well, I mean, that's one good reason. And the other reason is that it's been very well characterized. I mean, PTC has been studied since 1931. So, it's a very nice system, definitely.
But you're,
I'm sorry?
What is umami?
Well, yeah, it's that savory
question.
taste in meat. People
and
have,
MSG,
have
right?
have
What they
said that
add, the
MSG
additive that they
yeah.
often
Yeah.
put in. It's savory taste.
Yeah.
But I want to just follow up on your question about why this gene? And it is partly because it's the best worked out.
Yeah.
And it's a complex, the ability to taste, perception generally is pretty complex. And it's very rare that you actually will have one gene, one phenotype. And in this case, we could do that. But actually, there are many other substances that they're still trying to figure out what the genetic basis is of perception. It could be many genes, could be 10 genes, could be more than that.
Yeah. I mean, this is a very nice system because, I mean, it's very difficult to find a gene that correlates with a phenotype really, really strongly, or there are like 10 million genes that correlate with a particular phenotype. And this sort of like one gene, one phenotype is a nice way to demonstrate genotype-phenotype association.
Besides being able to detect toxins, are there any other known advantages to being able to taste PTC?
These receptors are expressed on the lung, and it's been found that when these receptors in the lung have been exposed to bitter chemicals, it will open the airways of the lungs and it will improve breathing. And so they're thinking that maybe that this could be a potential therapy for people who suffer from asthma.
Right, we don't know entirely. People used to think that these just had to do with taste, and it turns out they're wrong. Because they're expressed in the gut, they're expressed in the lungs. What is going on with these? There has to be something else going on, some other function. And that's an area of active investigation right now. People aren't entirely sure what the other functions may be.
Right.
And also keep in mind that we have the ability, people are classified as taster or non-taster for that particular substance, PTC. But that receptor potentially will recognize other chemicals, okay? And it has to do with sort of the lock and key that the chemicals fit into the receptor. And it's actually been hypothesized. Some people said, well, why is it that this non-taster haplotype is maintained? Why is that variant maintained and is so common? And some people thought, well, maybe it plays a role in perceiving another substance, not PTC. Right. Maybe there's something else out there that it plays a critical role in. And maybe it's not even taste, maybe it's some other physiologic phenomenon.
Yeah. And there's some anecdotal data that suggests that, the AVI haplotype actually recognizes another bitter substance as well. But we need to collect more evidence.
Is there anything that can happen to you, I guess, in an individual that would ruin your ability to taste the...
Yes. Not kidding, go.
Like, because I know it's a trait from the gene, but I mean, we burn our tongues, we do certain things. Can it take away your ability to actually taste it?
There is a difference. I mean, as you mentioned before, children are very sensitive to bitter substances. And as an adult, you become a little less sensitive. And as you get older, maybe, a little less sensitive. But if you're a taster, I mean, you're not suddenly going to become a non-taster. Like, it doesn't work that way. I mean, you might see a gradual decrease in ability, but you're not going to lose it completely.
Although generally there are factors that can influence, if you're having a lot of damage done to your tongue, and I think you were mentioning people who chew tobacco or
they
this other
temporarily
substance.
will dull their ability. Yeah.
But even if you damage, like say you burned your tongue, they grow back.
Yeah.
They'll regenerate the
will
cells,
grow
and
back,
so usually it won't have a long-term effect.
Yeah.
People at Monell Chemical Senses Center do a lot of testing in children. And it's well known, they've documented that bitter taste changes. It's definitely stronger in children. And then it weakens as you get older. And this actually has important implications for things like nutrition. And so toddlers are notoriously difficult to get to eat certain foods. And there may actually be now a genetic reason why they don't want to eat sprouts, right? And it also could influence their willingness and ability to take certain drugs. So the pharmaceutical industry is really, really interested in this as well. Right. So a colleague of mine is working at Monell, and she's going to try to test this hypothesis. She actually is looking at the gene expression in infants children and going on up in different developmental stages. Because her hypothesis, she thinks that the gene, these bitter taste receptors, are going to be downregulated as people get older. And she has to look at it, though, specifically in the place where they're expressed. You know, you have to do like a little biopsy, and then you have to actually measure the RNA expression. So RNA is made from DNA, and that's going to code for protein, and you would expect it to be higher in the cells if the gene's upregulated. She thinks that there may be downregulation of the bitter taste receptor genes as people age, and that's a hypothesis that she has, or at least earlier in development, like during childhood. You were next.
Oh, yeah.
As we learn more about PTC and other taste receptors, do you think that the food industry will find more advanced ways to synthesize flavors that influence our perceptions of food?
Yeah, there are definitely applications
They're
for this.
working on it.
Oh, yeah.
Nestle is one of the major
Nestle
funders of taste perception research.
Yeah.
There's others out there. And it's something, certainly the food industry is very interested in this, pharmaceutical industry is very interested in this as well. And now there's actually a study that came out about a year ago, and this is kind of a little bizarre and curious, which is that these bitter taste receptors aren't just expressed within your tongue.
Yeah.
They actually are expressed in your gut.
Yeah.
Okay, so you're actually sensing these foods actually in your intestine as you're digesting them. And the other thing is they're expressed in the lungs.
Yeah.
Why are they expressed in the lungs? And there's a lot we don't know. These must have another function. There must be something else going on as well.
And that could be part of the reason why we see them at such high frequency. It may not necessarily be related to diet, but because they have these other functions, they may be selected for based on that.
You were next.
What is the ultimate goal of your research, and how could your research end up influencing the medical industry?
That particular gene we were talking about, PTC, is of interest because, again, it affects perception, and that could affect pharmaceutical, whether you take drugs or not. But generally, genetics is important because there are genetic variants that influence drug response.
Yeah, exactly.
And so characterizing those variants will be critical for determining individual drug response. Mm-hmm. We have done that pretty well in Europeans, but not in Africans. Right. We know very little. And so if we want to get proper treatments, and we know people respond differently to drugs, we need to be characterizing that genetic variation.
Absolutely.
And you guys can ask any questions. You can ask us also about our careers and what we do and things like that.
Are you kidding?
Sure you can.
How much does the genetic theory that you teach and that you kind of learn in the classroom, how much does that underlie the practice of the experiments that you conduct?
Definitely classes help because there's some basic things that you really should understand when you're analyzing these data. I mean, first of all, it's important to know what a gene is. You have to understand transcription, translation, because once you understand the sequence, you then have to translate to proteins to sort of to do further analyses as well. So, I mean, it definitely plays a huge role.
And then the statistical analysis.
And then the statistical analyses as well. Right. So, I mean, statistics, yeah, wow. Yeah, statistics, a little bit of computational, analyses, so it all helps. It all comes together to help with the analyses and to make the analyses much stronger.
How did you guys get into the field of study you guys are in?
My undergraduate degree was in physical anthropology, and I was more interested in fossil hunting. And I thought, I'm going to go out and look for fossils. But then down the road, I became really interested in genetics. And then I just completely changed my focus, and I just started reading more about genetics, and I just found it so fascinating and interesting, and I took classes, and I was completely hooked. So my dissertation was focused on population genetics, and then Sarah was on my dissertation committee, and then I started working for her when I finished, and it's just been great ever since. And this focus on African genetic diversity has just been amazing. I mean, there's just so much phenotypic and genetic variation that you find, and there's just something new that you learn all the time when you're looking at these genomes. So it's been really exciting, at least, I mean, for me. I don't know how it's been for you.
And I guess what got me started was anthropology.
Yeah.
I actually read Margaret Mead, and she wrote a book on like Samoans and cultural anthropology. And I was just determined, I'm going to go to the field and do field work. And I went to Berkeley, and I was an anthropology major. And at Berkeley at the time, there was someone named Alan Wilson, who was a founder of molecular evolution. And so everybody was talking about this. How can we use genetics to learn more about human evolution? That inspired me to take genetics classes. And then I realized when I went to get my PhD, I better get really solid training in genetics if that's what I want to apply. So I went to Yale and got a PhD in genetics. But I've always combined those two interests together.
In a really good lab, so, yeah.
I also want to go to medical school. I want to be an anesthesiologist. And I wanted to know, when you're in class, is it, I know you have lectures and everything, but is it a lot of hands-on, like, or do you get to experience that a lot, or is it a lot of reading and, you know, taking notes and stuff? I know that that's an important part, but do you really get to understand, like, how you get to interact with certain people and things like that?
I mean, would you say it varies from program to program, university to university? I mean, my experience is, I mean, I did my undergrad in Canada. I'm Canadian. And at the University of Toronto, it was very, you know, classroom focused, very, you know, but I mean, you did have labs where you got some hands-on experience. I mean, we handled fossils, we, you know, did stuff like that. But I mean, it varies, it varies. It wasn't as, we didn't do as much practical stuff. But we did a lot more of the theoretical classroom kind of work. But I mean, it varies.
I do encourage you absolutely to do research as an undergraduate. And I could tell you that when we look into consider admissions for graduate school, for example, the undergraduate research is a critical factor. And so that's something to think of when you apply to colleges and where you go, that it's nice if you have an opportunity to work in labs. I have undergrads in my lab. I have them starting as freshmen all the way up to senior. And sometimes they'll just do internships, just watch. They just want to learn the methods. You have to learn if you like it. Yeah. You're not going to know if you like it until you're actually in there. And all this stuff sounds really glamorous when we get the conclusions, but the day-to-day stuff is a lot of hard work.
Yeah.
And so you won't know that until you're actually like in the lab and trying it, or, you know, like John, you have to be in the field digging for a while and see what you think of that. And also remember that, you know, you go to the place that is the best fit for you, and there can be a number of reasons for that. And very often it is, actually, my husband and I discussed this, he's an academic as well. Graduate school can actually be the really important thing down the road. So if you do decide to pursue a graduate degree, whether it be a PhD, or you're going to do an MD, you're going to go to law school, or whatever you're going to do, social work, whatever it is, that people down the road are going to care. It's going to matter more what you did actually for your PhD or for your graduate degree, I would say. And there are many opportunities. Even if you don't go to Columbia or an Ivy League school now, that doesn't at all rule out the possibilities down the road.
Yeah. I mean, I had an undergraduate working with me in Sarah's lab, and he survived. I mean, he told me he told me afterwards that I'm demanding. Sometimes I yelled at him.
No, you didn't.
Well, sometimes I did. But he was excellent. He was absolutely excellent. I mean, by the time he was done, he finished a project that was just totally amazing. And now he wants to pursue a PhD in evolutionary genetics.
And he's going to be a co-author on a paper.
A co-author on a paper, yeah, exactly. And I mean, it's tough and it's hard, but I agree with Sarah. I mean, definitely get in there and get into a lab, see if you like it, see if you can deal with the pressure and the day-to-day grime of, it's not always glamorous, of this kind of research, and then decide. But I mean, you want to be a medical doctor, so yeah. Medical doctors do research, so we'll see.
You have.
Do you know what kind of research project you might do next? Like if...
That's a good question.
What do you tell me?
I have no idea.
Well, what's the next stage in your career? So you're done a postdoc, and now?
I was going to stay in your lab forever.
No. He's applying for jobs. You're applying for faculty jobs, right?
Well, I'm looking, yeah.
And so that's the general progression.
That's
You do a
my
postdoc,
general.
You don't stay there forever, even though we'd love to have Michael.
She doesn't want to pay me for
But
it.
my job is to encourage the bird to leave the nest.
Yeah, that's true.
And to get a permanent faculty position is the long-term goal, and I'm sure he will be successful at that.
I hope so.
Do you ever find the monotonous tasks boring? Like, do you ever want to switch and go into just stay in the field? Or is it possible just to stay in the field?
Oh, yeah. Me, I like what I do, so I'm okay. I don't know about you.
It is, look, I'm going to be honest with you. The day-to-day lab stuff is really boring sometimes. It is, but what drives us is the excitement. It's when you find something,
When
you know,
you find
like
something, yeah,
when Michael
yeah.
found this result and he could look at, you know, it's sort of like you guys did that test, that taste test. And you can now, those of you who did the genetic test, you can look at that and see, wow, this like matches. I can see this correlation between my genotype and my phenotype, and it's sort of exciting. And when we see the result, but to get there, it is pretty tedious at times. And I think that's probably true of almost any field, probably.
Yeah. There'll be times when things are very slow and tedious and you don't want to do it. But I mean, things will happen and they'll remind you that, yeah, I really love what I'm doing and this is what I'm meant to do and this is really exciting. I think the only thing I don't like is lab meetings, and I think we should cut down on those things.
So what Michael's talking about, when you have a research lab, when you have like a genetics lab, you typically meet every week. You have a lab meeting. And so everybody in the lab has to get together and present their work, including you. But it's supposed to be a time where people can get, critical feedback, but in a positive way, and also share ideas. Sometimes if there's new papers, like we'll read the papers and things like that.
No.
So it's actually an important thing.
Yeah, but sometimes I want to write my own papers, and but anyway.
And I think we are pretty much out of time, so thank you so much for your participation.
Yeah, you guys, it's been great.